Hello everyone and welcome back to yet another video of the chemistry series. In this video we'll be doing the 14th chapter of chemistry, that is biomolecules. For the ones who are new here, we do NCERT and literally every single line of it in the least possible time, so you can watch your videos on the go without missing out anything. And especially for the ones who are, if your basics are done, it can be really helpful. So let's begin. A living system grows, sustains, and reproduces itself. The most amazing thing about a living system is that it is composed of non-living atoms and molecules. The pursuit of knowledge of what goes on chemically within a living system falls in the domain of biochemistry. Living systems are made up of various complex biomolecules like carbohydrates, proteins, nucleic acids, lipids, etc. Proteins and carbohydrates are essential constituents of our food. These biomolecules interact with each other and constitute the molecular logic of life processes. In addition, some simple molecules like vitamins and minerals, salts also play an important role in the functions of organisms. Structures and functions of some of these are discussed. So basically, if we go for the chemical aspect of what our biomolecules are made up of, what we are made of basically, so our food and everything. So relating biology and chemistry, that's what the first introductory paragraph is trying to say. So let's talk about carbohydrates, the first topic. Carbohydrates are primarily produced by plants and form a very large group of naturally occurring organic compounds. Some common examples of carbohydrates are cane sugar, glucose, starch, etc. इन सारे forms में carbohydrates present होते हैं और usually plants produce them, right? Most of them have a general formula that is this. So it's like hydrated carbon, ऐसा लेते हैं, ठीक है, CxH2OY times, and we're considered as hydrates of carbon. जो यहाँ पे लिख रहा है, from where the name carbohydrate was derived. For example, the molecular formula of glucose is C6H12O6. It can fit into this formula if you take out 6 carbon molecules out और water के 6 molecules बाहर. This will give you 6 times hydrated carbon. But all the compounds which fit into this formula may not be classified as carbohydrates. For example, acetic acid, it fits into this general formula, you can see, but it's not actually a carbohydrate. Similarly, rhamnose, it is a carbohydrate but does not fit in this definition. So these are basically the exceptions to what we call as the hydrated carbon theory of the carbohydrates. A large number of their reactions have shown that they contain specific functional groups. Chemically, the carbohydrates may be defined as optically active polyhydroxyaldehydes or ketones, or the compounds which produce such units on hydrolysis. So this is the chemical definition. के scientific point of view से ऐसे ऑप्टिकली एक्टिव पॉलीहाइड्रोक्सी मीन्स OH ग्रुप बहुत सारे एल्डिहाइड्स और कीटोन्स में हम अटैच कराएंगे एंड देर ऑप्टिकली एक्टिव विच मीन्स दे कैन एक्चुअली रोटेट द PPL विच वी हैव डन ऑलरेडी इन ऑर्गेनिक फर्स्ट लेसन. सो ऐसे कंपाउंड्स जो ऑप्टिकली एक्टिव होंगे, जो पॉलीहाइड्रोक्सी एल्डिहाइड्स या कीटोन्स होंगे या फिर हाइड्रोलिसिस पे वो इसके डेरिवेटिव्स प्रोड्यूस करें, उनको हम कार्बोहाइड्रेट्स बोलते हैं. सम ऑफ द कार्बोहाइड्रेट्स विच इज स्वीट इन टेस्ट आर ऑल्सो कॉल्ड एज शुगर्स. द मोस्ट कॉमन शुगर यूज्ड इन आवर होम्स इज सुक्रोज, जो हम यूज करते हैं, whereas the sugar present in milk is the lactose sugar. Carbohydrates are also called saccharides because saccharon means sugar. So saccharide, carbohydrates, one and the same thing. Then coming to classification of carbohydrates. So they're classified on the basis of their behavior on hydrolysis. हम उनको hydrolyze करेंगे, how will they behave? So they can be classified as mono, oligo, polysaccharides. ये terms आप mono, oligo, you can relate to them. You must have done them. Mono means one only. So which means इसमें सिर्फ एक ही saccharide group होगा. A carbohydrate that cannot be hydrolyzed any further to give simpler unit of polyhydroxyaldehyde or ketone is called a monosaccharide. About 20 monosaccharides are known to occur in nature. Some examples are glucose, fructose, ribose. You have to remember these. Monosaccharides आप उनको further नहीं तोड़ सकते उनके constituents में because वो already simplest form में होते हैं. Coming to oligosaccharides now. Oligosaccharides में these are those carbohydrates that yield 2 to 10 monosaccharide units. यानी वो कितने monosaccharide से बने हैं? 2 to 10. So on hydrolysis they'll give out such and such number of monosaccharide units, जिनको हम disaccharide, trisaccharide, tetrasaccharides भी बोलते हैं depending upon the number of monosaccharides they provide on hydrolysis. Amongst these, the most common are disaccharides, जो बहुत common थे. हम biology में usually we talk about those only. The 2 monosaccharide units obtained on hydrolysis of a disaccharide may be different or same. ठीक है, तो disaccharide के जब आप 2 monosaccharide वाले units मिलेंगे, वो same भी हो सकते हैं, different भी हो सकते हैं. Say one molecule of sucrose, very important. On hydrolysis, it gives glucose and fructose. So this sucrose is a disaccharide, gives out glucose and fructose, जो दोनों monosaccharides है, जबकि maltose जो only gives 2 molecules of glucose. Similar monosaccharide units. अब polysaccharides क्या होंगे? Which will yield a large number of monosaccharide units on hydrolysis, उनको polysaccharides बोलते हैं. एग्जाम्पल स्टार्च, सेल्यूलोज, ग्लाइकोजन जिसमें हम अपना फूड स्टोर करते हैं, गम्स एक्सेट्रा. फाइन, these are all examples of polysaccharides. Polysaccharides are not sweet in taste, hence they are also called non-sugars. What's the reason for them being called as non-sugars? Because they do not taste sweet. तो हमारे लिए usually for a layman sugar is something that is sweet. So polysaccharides are non-sugars because they're not sweet and they yield large number of monosaccharides when they're hydrolyzed. The carbohydrates may be also classified as either reducing or non-reducing sugars. All right, अब एक और classification आता है, या तो reducing या फिर non-reducing. All those carbohydrates which reduce the Fehling solution and Tollens reagent, we have done them already. Organic में आते हैं, बहुत important reagents हैं, इनसे हम टेस्ट करते हैं aldehydes वगैरह का, referred to as reducing sugars. ठीक है, जो Fehling solution, Tollens reagent को reduce करेंगे उनको reducing sugar, और जो नहीं करेंगे all monosaccharides whether aldose or ketose are reducing sugars. ये सारे reduce करते हैं इन reagents को. Then we have monosaccharides. They further classified on the basis of number of carbon atoms and the functional group present in them. If a monosaccharide contains an aldehyde group, it is known as aldose, and if it contains a keto group, it is known as a ketose. हमने already देखा, अब हमारे पास जो हमारे carbohydrates होते हैं वो या तो पॉलीहाइड्रोक्सी एल्डिहाइड्स है या फिर पॉलीहाइड्रोक्सी कीटोन्स है. तो अब अगर उनमें एल्डिहाइड ग्रुप है तो एल्डोज बोलेंगे, कीटो ग्रुप है या तो हम उसको कीटोज बोलेंगे. नंबर ऑफ कार्बन एटम्स कॉन्स्टिट्यूटिंग द मोनोसैकेराइड इज ऑल्सो इंट्रोड्यूस्ड इन द नेम एज इज एविडेंट फ्रॉम द एग्जाम्पल्स गिवन बिलो. तो कितने मोनोसैकेराइड में कितने नंबर ऑफ कार्बन, जैसे यू हैव ट्रायोज, हाँ, एल्डो ट्रायोज, कीटो ट्रायोज. देन यू हैव टेट्रोज 4, वेरी इजी, 5 पेंटोज, 6 हेक्सोस, 7 हेप्टोज. तो एल्डो कीटो लगाएंगे साथ साथ इफ इट्स अटैच्ड टू एन एल्डिहाइड ग्रुप और अटैच्ड टू अ कीटोन ग्रुप. ग्लूकोज वेरी इंपॉर्टेंट. अब इसका आज हम प्रिपरेशन करेंगे. आप यू मस्ट हैव हर्ड अबाउट ग्लूकोज समवेयर, कहीं ना कहीं तो पढ़ा ही होगा बायो में. ग्लूकोज occurs freely in nature as well as in the combined form. It is present in sweet fruits and honey. Ripe grapes also contain glucose in large amounts. And how is it prepared? From sucrose और cane sugar जो है हमारा disaccharide, that is sucrose. उसको जब हम boil करते हैं with dilute HCl acid में और H2SO4 in alcoholic solution, glucose and fructose are obtained. तो हम basically यहाँ पे क्या कर रहे हैं, हम इसको तोड़ रहे हैं इसके monomeric unit में, और हमें पता है sucrose gives glucose and fructose, and that's how you get glucose from cane sugar. यहाँ पे दिया हुआ this is sucrose, C12H22O11, and this is giving you glucose and fructose. उसके बाद starch है हमारे पास. Commercially glucose is obtained by hydrolysis of starch by boiling it with dilute H2SO4. तो कुछ भी आपने acidic medium दे दिया, temperature दे दिया, boil कराया, हमारा starch और cellulose is a polysaccharide, and this will give you n times glucose. Alright, what is now the structure of glucose? ये भी आपने किया ही होगा, I'm sure. This is the structure of glucose. ठीक है, ये यहाँ पे हमारे ग्लूकोज इज एन एल्डोहेक्सोस, हाँ, 6 कार्बन है और एल्डिहाइड ग्रुप अटैच्ड है, एंड इट इज ऑल्सो नोन एज डेक्सट्रोज. इट इज द मोनोमर ऑफ मेनी ऑफ द लार्जर कार्बोहाइड्रेट्स नेमली स्टार्च, सेल्यूलोज. ये सब किसके बने हैं? ग्लूकोज के बने. इट इज प्रॉब्ली द मोस्ट अबंडेंट ऑर्गेनिक कंपाउंड ऑन अर्थ. इट वाज असाइंड द स्ट्रक्चर गिवन बिलो ऑन द बेसिस ऑफ द फॉलोइंग एविडेंसेस. तो ये 4 दफे रिपीट हो रहा है. दिस इज द 5th कार्बन एंड दिस इज द 6th कार्बन. ऑलराइट, ये हमारा पॉलीहाइड्रोक्सी एल्डिहाइड है, ऊपर हमारा एल्डिहाइड है और hydroxyl groups बहुत सारे. Its molecular formula was found to be this C6H12O6. We all know this. Photosynthesis का formula सबने किया है, I believe. On prolonged heating with HI, it forms N-hexane, suggesting that all the 6 carbons are linked in a straight chain. यानी कोई cyclic compound नहीं है. जब हम इसको HI के साथ react करवाते हैं तो हमें वहाँ से N-hexane मिलता है. Glucose reacts with hydroxylamine. All right. This is hydroxylamine NH2OH to form an oxime and adds a molecule of hydrogen cyanide to give cyanohydrin. These reactions confirm the presence of a carbonyl group in glucose. So there's a carbonyl group क्योंकि aldehyde है ऊपर. जब आपने इसको hydroxylamine के साथ react करवाया, you got an oxime. This is an oxime. Alright, और इसको बाद में what did you do? HCN cyanide के साथ react करवाया and you got cyanohydrin. ठीक है, ऊपर वाले जो H था, जो O था, it got replaced by this CnNOH. Glucose gets oxidized to 6 carbon carboxylic acids. Gluconic acid जिसको बोलते हैं. जब आप ग्लूकोज aldehyde है, when you oxidize it, it will get converted into carboxylic acid. On reaction with a mild oxidizing agent like bromine water, हाँ, mild oxidizing agent लेंगे, alright, और हमें मिलेगा हमारा carboxylic acid from the aldehyde. Fine. ये ऑक्सीडेशन रिडक्शन हमारे कन्वर्जन सीरीज होते हैं which we have done in organic chemistry. Carbonyl group is present in as an aldehyde group. Acetylation एसिटिक एनहाइड्राइड ऐड कराया है, ठीक है, ऑफ ग्लूकोज विद एसिटिक एनहाइड्राइड गिव्स ग्लूकोज पेंटा एसिटेट विच कंफर्म्स द प्रेजेंस ऑफ 5 OH ग्रुप्स. पेंटा एसिटेट मतलब 5 दफे 5 OH के साथ क्या मिला है, एसिटिक एनहाइड्राइड मिला है. इसका मतलब हमें पेंटा एसिटेट मिलता है विच कंफर्म्स द प्रेजेंस ऑफ 5 OH हाइड्रोक्सिल ग्रुप्स इन द ग्लूकोज मॉलिक्यूल. क्योंकि छठे कार्बन पे फिर OH नहीं होता, वहां पे एल्डिहाइड ग्रुप होता है. सिंस इट एग्जिस्ट एज अ स्टेबल कंपाउंड 5 OH group should be attached in different carbon atoms. एक ही कार्बन पे 2 H होते हैं, इट्स अनस्टेबल. ऑन ऑक्सीडेशन विद नाइट्रिक एसिड नाउ, ठीक है, अब ऑक्सीडाइज करेंगे तो हमें पता है हमें एसिड्स मिलता है. ग्लूकोज एज वेल एज ग्लूकोनिक एसिड बोथ यील्ड a dicarboxylic acid, saccharic acid. This indicates the presence of a primary alcoholic group in glucose. अब यहाँ पे ये क्या कह रहे हैं, हमने नाइट्रिक एसिड के साथ ऑक्सीडेशन कराया और यहाँ पे सैकरिक एसिड मिला हमें विच इज़ बेसिकली अ डाइकार्बोक्सिलिक एसिड, 2 कार्बोक्सिलिक ग्रुप्स आए. ऑलराइट, और यहाँ पे अगेन ग्लूकोनिक एसिड लें आप, और वेदर यू टेक ग्लूकोज बोथ आर गोइंग टू यील्ड द सेम. इससे पता चलता है कि एक प्राइमरी अल्कोहलिक ग्रुप है ग्लूकोज में जो बेसिकली ऑक्सीडाइज होके हमको 2 carboxylic groups there. The exact spatial arrangement of different OH groups was given by Fischer after studying many other properties. Its configuration is correctly represented as first, so gluconic acid is represented as second and saccharic as third. So ये हमारा actually this is glucose basically. Then you have your the second one is gluconic acid and third one is saccharic acid. Glucose is correctly named D plus glucose, ठीक है, D dextro basically. D before the name of glucose represents the configuration, whereas plus represents dextro rotatory nature of the molecule. Dextro मतलब right की ओर, levo we know it's leftwards. It should be remembered that D and N have no relation with the optical activity of the compound. They're also not related to letter D and L. The meaning of D and L notations is as follows. यहाँ पे D और L का क्या मतलब है? The letters D or L before the name of any compound indicate the relative configuration of a particular stereoisomer of a compound with respect to configuration of some other compound. हम यहाँ पे stereoisomerism देख रहे हैं, spatial arrangement के difference पे जो isomerism दिखाते हैं compounds, ठीक है. तो यहाँ पे plus would mean right side से हमारा OH group है और minus would mean कि left side पे OH group है. It's just the spatial arrangement of this hydroxyl group. And it has nothing to do with the rotatory activity. In the case of carbohydrates, this refers to the relation with a particular isomer of glyceraldehyde. Glyceraldehyde contains one asymmetric carbon atom and exists in 2 enantiomeric forms. Enantiomers, non-superimposable mirror images of each other, although they're same. ये isomeric है. This is glyceraldehyde. Isomer, plus वाला isomer, it has D configuration. It means that when its structural formula is written on paper, following specific conventions which we'll study in high class, the OH group lies on the right-hand side. जो already हमने देखा, glyceraldehyde plus वाले में OH will be on the right side. All those compounds which can be chemically correlated to D plus isomer of glyceraldehyde are said to have D configuration, whereas those which can be correlated to L or minus isomer of glyceraldehyde are said to have L configuration, जिनमें left side पे होगा. जो हमारा एक बीच वाला asymmetric carbon होता है उसमें 2 possibilities आता है, either it's D or it's L. Alright. L or minus isomer of OH group is on left hand side, as you can see in the structure. For assigning the configuration of monosaccharides, it is the lowest asymmetric carbon atom which is compared. ठीक, एज इन प्लस ग्लूकोज OH ऑन द लोवेस्ट एसिमेट्रिक, पहले सबसे कम नंबरिंग जिसको मिली है, बिकॉज अब यहाँ पे बहुत सारे OH ग्रुप्स है, जो सबसे कम वाली नंबरिंग वाला OH ग्रुप होगा हम उसका जो सिमेट्री है वो कंसिडर करेंगे ऑन द राइट साइड विच इज कंपेरेबल टू प्लस ग्लिसरल्डेहाइड. सो प्लस ग्लूकोज इज असाइन टू D कॉन्फिगरेशन, जहाँ OH ग्रुप राइट साइड पे होगा, किस कार्बन पे, एसिमेट्रिक कार्बन पे. अब इसमें बहुत सारे एसिमेट्रिक कार्बन है, जो लोएस्ट नंबर वाला एसिमेट्रिक कार्बन है वो वाला लेंगे. अदर एसिमेट्रिक कार्बन्स ऑफ ग्लूकोज आर नॉट कंसिडर्ड फॉर दिस कंपैरिजन. तो सारे टेढ़े हो जाएंगे. Also, the structure of glucose and glyceraldehyde is written in a way that most oxidized carbon, in this case CHO, is at the top. Most oxidized carbon हमें पता है aldehyde group में ही अभी so far in this structure aldehyde में ही सबसे ज़्यादा oxidation हुआ है, right? ये हमारा glyceraldehyde दिया हुआ है और ये हमारा D glucose, dextroglucose. plus sign indicates कि OH group कहाँ होगा, right side पे होने वाला है. Clear? साइक्लिक स्ट्रक्चर ऑफ ग्लूकोज. अब ग्लूकोज का साइक्लिक स्ट्रक्चर कैसा होगा? द स्ट्रक्चर फर्स्ट जो हमने अभी किया, एक्सप्लेन मोस्ट ऑफ इट्स प्रॉपर्टीज, बट द फॉलोइंग रिएक्शंस एंड फैक्ट्स कुड नॉट बी एक्सप्लेन बाय द स्ट्रक्चर. कुछ चीज़ें थी विच वाज स्टिल देयर वाज अ डिलेमा कि हम बता नहीं सके कि क्या है ग्लूकोज का प्रॉपर्टी, सो वी हैड टू कंसिडर द साइक्लिक स्ट्रक्चर. डिस्पाइट हैविंग द एल्डिहाइड ग्रुप, ग्लूकोज डज नॉट गिव शिफ्ट्स टेस्ट एंड इट डज नॉट फॉर्म द हाइड्रोजन सल्फाइड एडिशन प्रोडक्ट विद NaHSO3. Now we know that shift test is a chemical test. It's used to check for the presence of aldehydes in a given analyte. All right. तो वो भी ये नहीं देता in its open structure. The penta-acetate glucose of glucose does not react with hydroxylamine, indicating the absence of free CHO group. Glucose is found to exist in two different crystalline forms, which are named as alpha and beta. The alpha form of glucose, हाँ आपने इससे पहले भी कभी देखा होगा किसी डाइसैकेराइड के दो मोनोसैकेराइड यूनिट्स है. You get alpha form and you get beta form. Alpha wala is obtained by crystallization. Just a second. Alright, alpha form of glucose is obtained by crystallization from concentrated solution of glucose, while the beta form is obtained by crystallization from hot and saturated aqueous solution. So you have two different forms. This behavior could not be explained by the open chain structure for glucose. It was proposed that one of the OH groups may add to the CHO and form a cyclic hemiacetal structure. एक OH जो है CHO के साथ जोड़ के क्या बनाएगा? Hemiacetal structure बनाएगा. It was found that glucose forms a six-membered ring in which OH at fifth carbon is involved in a ring formation. पांचवें कार्बन ये हमारा फिफ्थ कार्बन है. दिस विल बी इन्वॉल्व. इसका OH जो है वही बनाएगा हेमियासिटल. दिस एक्सप्लेन्स द एब्सेंस ऑफ CHO ग्रुप. एंड ऑल्सो अब CHO गायब हो गया तो अब हम ये भी बता सकते हैं कि Schiff's test नहीं देगा, अब देगा. एंड ग्रुप एंड ऑल्सो एक्सिस्टेंस ऑफ ग्लूकोज इन टू फॉर्म्स एज शोन बिलो. अब हम देखेंगे यहाँ पे अल्फा और बीटा में क्या डिफरेंस है. आप अल्फा बीटा ओपन चेन स्ट्रक्चर में आप नहीं डिमार्केट कर सकते हैं बिकॉज वहाँ पे दोनों सेम ही होते हैं. यहाँ पे सी अल्फा एंड बीटा. अल्फा के केस में यू कैन सी यहाँ पे फर्स्ट कार्बन पे जो OH है वो राइट में है इन केस ऑफ अल्फा, एंड इन केस ऑफ बीटा व्हाट डू वी हैव इज लेफ्ट पर है. बाकी सारा सेम है. ऑलराइट, सो दैट्स हाउ यू कैन डू इट. द टू साइक्लिक हेमियासिटल फॉर्म्स ऑफ ग्लूकोज डिफर ओनली in the configuration of the hydroxyl group at carbon 1 called the anomeric carbon. The aldehyde carbon before cyclization. Cyclization से पहले जो कार्बन aldehyde वाला कार्बन था CHO बना रहा था उसको anomeric carbon बोलते हैं. उस कार्बन के साथ जो hydroxyl group का जो configuration होगा, whether it's on the left side then it will be the beta glucose, if it's on the right side it is the alpha glucose. Such isomers, that is the alpha form and beta form, they call anomers. Anomers, anomeric carbon में difference था. The six-membered cyclic structure of glucose is called pyranose structure. Alpha or beta and analogy with pyran. Pyran ring, this is a pyran ring, तो इसलिए इसको pyranose बोलते हैं because ये resemble करता है. Pyran is a cyclic organic compound with one oxygen atom and 5 carbon atoms in the ring. यहाँ पे भी same है, 1 oxygen 5 carbon. The cyclic structure of glucose is more correctly represented by the Haworth structure as given below. This is आप usually ऐसे देखेंगे. जो हमने ऊपर cyclic structure देखा, you don't see glucose like this. Usually you see it like this. Alright, so this is the alpha glucopyranose. Why pyranose? Because ये पायरन रिंग को रिजेंबल करता है. पायरन रिंग में 5 कार्बन और एक ऑक्सीजन होता है. बीटा में अगेन जो है लेफ्ट साइड पे होगा, अल्फा में राइट साइड पे होगा. आपने ऊपर वाले स्ट्रक्चर में देखा था. यहाँ यू कैन सी जो फर्स्ट कार्बन है जो हमारा एनोमेरिक कार्बन है OH नीचे से है अल्फा में और बीटा के केस में ऊपर है. कमिंग टू फ्रक्टोज नाउ. फ्रक्टोज जो है इज एन इम्पॉर्टेंट कीटोहेक्सोस. अभी तक हम एल्डोहेक्सोस पढ़ रहे थे इन केस ऑफ ग्लूकोज. इट्स ऑब्टेन्ड along with glucose by the hydrolysis of disaccharide sucrose. Alright, it's a natural monosaccharide found in fruits, honey and vegetables. In its pure form it's used as a sweetener. It is also an important ketohexose. Now what is the structure? अब ऊपर कीटोन ग्रुप लगेगा और कीटोन्स जो होते हैं वो टर्मिनल नहीं होते हैं, तो एक हाइड्रोक्सिल वाला कार्बन इसके ऊपर भी होगा. फ्रक्टोज also has the molecular formula same, C6H12O6 as glucose, and on the basis of its reactions was found to contain a ketonic functional group at carbon number 2. ठीक है, and 6 carbons in straight chain as in the case of glucose. It belongs to D series and is a levorotatory compound. ठीक है, अब यहाँ पे dextro levo different है. D series just means कि what is a position, और levorotatory is something else. It's appropriately written D minus और ये अलग से minus यहाँ पे. ठीक, minus का क्या मतलब है? वही levorotatory. Its open structure is as shown. It also exists in two cyclic forms which are obtained by the addition of OH at C5 to the carbonyl group. The ring thus formed is a 5-membered ring and is named as furanose. अब furan ring क्या होता है with analogy to the compound furan? Furan is again a 5-membered ring with 1 oxygen and 4 carbon atoms. ठीक है, pyran ring was something else. यहाँ पे 5 कार्बन और एक ऑक्सीजन था, यहाँ पे 4 कार्बन और एक ऑक्सीजन है हमारे पास. This is the furan ring. तो furanose बोलते हैं कीटोस शुगर को. Why? Because resemblance है. So you can have over here alpha वाला और बीटा वाला. Alright, so alpha में एज वी नो जो अल्फा और बीटा है उनमें जो हमारा एनोमेरिक कार्बन होता है उनमें हाइड्रोक्सिल का पोजीशन डिफरेंट होता है. सो दैट्स हाउ यू डू इट. और यूजुअली हमारे हैवर्थ स्ट्रक्चर्स वही आते हैं. दिस इज यू टू रिमेंबर दिस वन. साइक्लिक स्ट्रक्चर्स ऑफ 2 anomers of fructose are represented by Haworth structures. You can see this is fructofuranose, बीटा वाला एंड अल्फा वाला. फाइन, सो दैट्स हाउ योर फ्यूरन रिंग लुक्स लाइक. और यहाँ नीचे होगा अल्फा में और बीटा में ऊपर होगा हाइड्रोक्सिल, बाकी सारा सेम है. ऑलराइट, Okay. Now, what is disaccharide? You've already read that disaccharides on hydrolysis with dilute acids or enzymes yield two molecules of either the same or different monosaccharides. हमने examples भी देखे थे. The two monosaccharides are joined together by an oxide linkage formed by the loss of a water molecule. ये दो आपस में linked होंगे. अगर आप H2O बाहर निकालते हैं तो oxygen से दोनों monosaccharides can be linked. Such a linkage between two monosaccharide units through the oxygen atom is called glycosidic linkage. Very important. Disaccharide two tied into two monosaccharides. अगर वो linked है oxygen से तो हम उसको बोलेंगे glycosidic linkage. In disaccharides, if the reducing groups of monosaccharides, that is aldehydic or ketonic groups, are bonded, these are non-reducing sugars. Example, sucrose. On the other hand, sugars in which the functional groups are free are called reducing sugars, जो दूसरों को reduce कर सकते हैं. ठीक है. So, for example, maltose and lactose. Alright, sucrose. One of the common disaccharides is sucrose, which on hydrolysis gives equimolar mixture of dextro वाला plus glucose and minus fructose. ठीक, यहाँ पे sucrose दिया हुआ है water molecule के साथ. What happens is you will get glucose and fructose. These two monosaccharides are held together by a glycosidic linkage between carbon number 1 of alpha glucose and carbon number 2 of beta fructose. Since the reducing groups of glucose and fructose are involved in glycosidic bond formation, sucrose is a non-reducing sugar. Reducing groups जो थे वो बंध चुके हैं, उनका linkage हो चुका है. As a result, वो अब non-reducing sugar बन जाएगा. So sucrose is a non-reducing sugar. What makes you say that? Because the reducing groups are already involved in glycosidic linkage. अब वो free नहीं है. If the reducing groups are not free, the aldehyde, the ketone group is not free, so you can say that it is a non-reducing sugar. Sucrose is dextrorotatory. हमने already देखा, but after hydrolysis gives dextrorotatory glucose and levorotatory fructose. Since the levorotation of fructose is more than the dextrorotation of glucose, the mixture is levorotatory. Thus, hydrolysis of sucrose brings about a change in the sign of rotation from dextro to levo, and the product is named as invert sugar. Inversion क्यों हुआ? Sucrose dextro था. Hydrolysis के बाद हमें major compound जो है levorotatory मिला, so we call it a levo product eventually, and इसी sugar को invert sugar बोलते हैं. Coming to maltose, it's another disaccharide. Maltose is composed of 2-alpha-D-glucose, in which carbon number 1 of glucose first is linked to carbon number 4 of another glucose unit. ठीक है, पहले का carbon number 1, दूसरे का carbon number 4. ये आपस में linkage दिखा रहे हैं. The free aldehyde group can be produced by carbon number 1 of second glucose in solution, and it shows reducing properties, so it's a reducing sugar. Why is maltose a reducing sugar? Now what makes you say that? Very easy. हमने एक glucose निकला, दो glucose molecules निकले maltose से, alpha D glucose. They were linked. एक का carbon 1, एक का कार्बन 4, और यहाँ पे हमने क्या देखा, इसका जो हमारे पास एल्डिहाइडिक ग्रुप था, that was still there. Fine, you can see it over here. हमारा जो एल्डिहाइड है वो कहीं नहीं गया ग्लूकोज का, तो still it has reducing properties because this is still free. I'm sorry, this is the hydroxyl group. This is your basically the free aldehyde group. जो आपका CHO group है वो यहाँ से free होगा, इसका 4th कार्बन, because इस वाले का लिंक हो रहा है. Then we have lactose. ये एक और disaccharide है. It's more commonly known as milk sugar. It's found in milk. आपने सुना होगा. It's composed of beta D galactose and beta D glucose. Lactose को तोड़ेंगे तो galactose और glucose मिलेगा. बायो में भी आप करते हैं. Linkage is between the carbon number 1 of galactose. ठीक है, galactose का first carbon जाएगा and 4th carbon of the glucose. ठीक. Again, you can see a free aldehyde group. It may be produced at the carbon number 1 of glucose unit. Hence, it's also a reducing sugar. अभी भी हमारे पास CHO group जो है is still present over there. So we can say that हर एक का इसका अगर गया पहला वाला, इसका नहीं गया. So you can say that lactose is again a reducing sugar just like maltose. Why was it so? This is so because आपका जो aldehydic group है it's still free. Then we have polysaccharides. Contain a large number of monosaccharide units joined together by glycosidic linkages. ठीक है, अब हमें समझ आया glycosidic linkage क्या होता है. बीच में ऑक्सीजन होगा, दो monomeric units को link करेगा. These are the most commonly encountered carbohydrates in nature. They mainly act as the food storage or structural material. This is starch, glycogen, that's food storage. Starch is the main storage polysaccharide of plants. ये आपने पढ़ा होगा, glycogen animals में होता है, plants में starch होता है. It's the most important dietary source for human beings. High content of starch is found in cereals, root, tubers, some vegetables. A polymer of alpha glucose and consists of two components, amylose and amylopectin. तो amylose होगा और amylopectin होगा हमारे starch में. Alright. Alright, so amylose is water-soluble component, constitutes about 15 to 20% of starch. Chemically, amylose is a long unbranched chain of 200 to 1000 alpha D-glucose units held together by carbon 1, carbon 4 glycosidic linkage. ठीक है, amylopectin जो है on the other hand, it is insoluble in water, constitutes about 80 to 85% of starch. Major component amylopectin होगा, it's branch chain polymer of alpha D-glucose units in which chain is formed by C1, C4 glycosidic linkage, whereas branching occurs by C1, C6 glycosidic linkage. ये हमारा amylose है, हमारे पास alpha 1,4 linkages हैं. These are all the glucose molecules that we have, alpha D glucose molecules. Amylopectin, the insoluble component, 80% of starch. ये जो है 1 से 6th carbon में जो है branching हो रही है. You can see these are all branches. वरना in a line there's again 1,4 linkage only. You can see this is the alpha linkage, 1,4 linkage, but branching भी है तो that occurs between 1 and 6. वो ऊपर वाला paragraph बोल रहा था और लिया आपने सब में glucose units ही है. Coming to cellulose now, occurs extensively in plants, the most abundant organic substance in plant kingdom. ठीक है, most abundant organic substance, a predominant constituent of cell wall. Cellulose is a straight chain polysaccharide composed of only beta D glucose units, a homopolymer of glucose cellulose, ठीक है, which are joined by glycosidic linkage between carbon 1 of one glucose unit and carbon 4 of the next glucose unit. Usually carbon ऐसे जब हम glucose में देखते हैं तो 1,4 glycosidic linkage होता है and it's a homopolymer. You will see these are the beta links. Beta link क्यों? क्योंकि हमारे पास beta glucose molecules हैं. ऊपर amylose वगैरह में alpha links थे. This is cellulose और हमें पता है cell wall में present होता है. Glycogen, the carbohydrates are stored in the animal body as glycogen. Plants store it as starch. It's also known as animal starch because its structure is similar to amylopectin and is rather more highly branched. Branching थोड़ी ज़्यादा है, but glycogen is similar to amylopectin. It is present in liver, muscles and brain, जहाँ हमारी body इसको store करती है. When the body needs glucose, enzymes will break the glycogen down to glucose. You do not supply 24/7 glucose to your body. So what the body does is, if it requires it, it will break down the glycogen. It has specific enzymes to do so, and you'll get glucose. Glycogen is also found in yeast and fungi, not just the animal bodies. Coming to importance of carbohydrates, they're essential for life in both plants and animals. They form a major portion of our food. We know it. Honey has been used for a long time as an instant source of energy by the Veds. In the Ayurvedic system of medicine, carbohydrates are used as storage molecules, as starch in plants and glycogen in animals. I don't think we need to explain this anymore. We know it all. Carbohydrates form the basis of our food habits and everything. Whatever we eat mostly comprises of carbohydrates. Cell wall of bacteria and plants is made up of cellulose only. We build furniture etc from cellulose in the form of wood and clothe ourselves with cellulose in the form of cotton fiber. ये सारा cellulose है. They provide raw materials for many important industries like textiles, paper, lacquers and breweries. 2-aldopentoses, ribose, 2-deoxy-D-ribose, we know it already. ये nucleic acids में होते हैं. RNA, DNA. Carbohydrates are formed in biosystem in combination with many proteins and lipids. हमारी cells में present होता है. हमारे organelles carbohydrates से बने, हम खाते carbohydrates हैं. Everything, it's all about carbohydrates. So storage इससे होता है, बहुत सारे raw materials provide करता है. So that's it. Coming to now proteins. Proteins are the most abundant biomolecules of the living system. Chief sources of proteins are milk, cheese, pulses, peanuts, fish, meat, etc. They occur in every part of the body and form the fundamental basis of structure and functions of life. Bodybuilding होते हैं, we know it. Proteins are bodybuilding. They're also required for growth and maintenance of body. The word protein is derived from Greek word yos, which means primary or of prime importance. जो primary हमारी body में primarily हमारी body को ज़रूरत होते हैं वो proteins हैं. And all proteins are polymers of alpha amino acids. Very important. Amino acid आपको पता था. Another thing that you have to remember is it's not just amino acid, it is alpha amino acid जिसमें हमारी body के सारे proteins जो है आते हैं. What are now amino acids? Let's come to the monomeric unit of proteins. Amino acids contain the amine group. Acid है तो carboxyl group भी होगा. ये functional groups होते हैं. You're given an alpha amino acid. ठीक है. R is the side chain. You can see this alkyl group can be anything depending upon the relative position of amino group with respect to the carboxyl group. The amino acids can be alpha, beta, gamma, delta and so on. Carboxylic acid group के respect में amino group को कहाँ रखा है? यहाँ alpha position पे ही रखा है, right next to it. This is alpha amino acid. Only alpha amino acids are obtained on hydrolysis of proteins. इसका मतलब proteins सिर्फ alpha amino acid के बने हैं. They may contain other functional groups as well. All alpha amino acids have trivial names which usually reflect the property of that compound or its source. Glycine is so named. हर एक का अपना अपना नाम है. बायो में भी there's this chapter जहाँ पे आपको different नाम पता चलते हैं based upon उनके पास alkyl group कौन सा होता है. Glycine is so named it has sweet taste. Glycos means sweet. Triosine first obtained from cheese. Tyrose means cheese. Amino acids are generally represented by a 3-letter symbol. Sometimes 1-letter symbol is also used. Structure of some commonly occurring amino acids with their 3-letter and 1-letter symbols are given. Usually 3 letters का आपने देखा होगा ना, glycine, alanine, valine, leucine. ये सारे जो है इनमें साइड चेन जो आर है. ग्लाइसिन में इट्स जस्ट एच, एलेनिन में इट्स द मिथाइल ग्रुप, वैलिन में ये है, ल्यूसिन में ये है. ये आपके पहले लेटर्स वाले एक एक एब्रिविएशन है. देन यू हैव आइसोल्यूसिन, यू हैव आर्जिनिन, वेरी इंपॉर्टेंट. ठीक है, आर्जिनिन में क्या आप देख रहे हैं, लाइसिन अगेन वेरी इंपॉर्टेंट. जिन पे स्टार मार्क है वो ऑटोमेटिकली इंपॉर्टेंट बनते हैं. वी हैव थ्रियोनिन अगेन, then we have methionine, very important. Phenylalanine, tyrosine, ये सारे आपके amino acids. You just go through the table. Tryptophan, histidine, proline. Essential amino acid A, entire structure. Alright, now classification of amino acids. They are classified as acidic, basic or neutral depending upon the relative number of amino acid and carboxylic groups in their molecule. अगर जो है एसिडिक जो हमारे कार्बोक्सिलिक एसिड ग्रुप्स है वो ज़्यादा है, इट्स एसिडिक. अमीनो ग्रुप ज़्यादा है, बेसिक. इक्वल है तो न्यूट्रल. इजी. इक्वल नंबर ऑफ अमीनो एंड कार्बोक्सिल ग्रुप्स मेक्स इट न्यूट्रल. मोर नंबर ऑफ अमीनो दैन कार्बोक्सिल मेक्स इट बेसिक, एंड मोर कार्बोक्सिल ग्रुप्स एज कंपेयर टू अमीनो ग्रुप्स मेक्स इट एसिडिक. द अमीनो एसिड्स विच कैन बी सिंथेसाइज्ड इन द बॉडी आर नोन एज नॉन एसेंशियल अमीनो एसिड्स. जो हमारी बॉडी खुद बना सकती है उनको हम नॉन एसेंशियल बोलते हैं. ऑन द अदर हैंड दोज विच कैन नॉट बी सिंथेसाइज्ड दे मस्ट बी ऑब्टेन थ्रू द डाइट. जो हमारी बॉडी खुद नहीं बनाती है वो उसको बाहर से देना है. दे नोन एज एसेंशियल अमीनो एसिड्स. तो अब एसेंशियल नॉन एसेंशियल के एग्जांपल्स भी याद रखने हैं. दे आल्सो कम इन एग्जाम. अमीनो एसिड्स आर यूजुअली कलरलेस क्रिस्टलाइन सॉलिड्स. दीज आर वाटर soluble high melting solids and behave like salts rather than simple amines or carboxylic acids. तो usually आप अगर इनको ऐसे you might be thinking about them as amines, as basic compounds, but they're usually salty, fine in appearance. This behavior is due to the presence of both acid and basic group in the same molecule. In aqueous solution, the carboxyl group can lose a proton and amino can accept, giving rise to a dipolar ion known as Zwitter ion. This is neutral but contains both positive and negative charges. यहाँ पे आप देखेंगे aqueous solution में क्या होगा, इसका इलेक्ट्रॉन जो है वो इस ऑक्सीजन पे चला जाएगा, इवेंचुअली यहाँ से हाइड्रोजन रिमूव हो जाएगा और हमारे पास Zwitter ion आता है. तो it's neutral yet it contains charges in the aqueous solution. Amino acids show amphoteric behavior as they react both with acids and bases. Acid से भी कर सकते हैं, base से भी. तो Zwitter ionic form is usually the amphoteric form. Except glycine, all other naturally occurring alpha amino acids are optically active since the alpha carbon atom is asymmetric. अभी glycine में हमने देखा हाइड्रोजन ही आ जाता है. ठीक है, तो सो यहाँ पे जब दो सिमिलर ग्रुप्स आते हैं तो वी नो ऑप्टिकली इनएक्टिव हो जाता है कंपाउंड. लेकिन अगर हमारे पास एसिमेट्रिक कायरल कार्बन होगा जिसमें सारे ग्रुप्स डिफरेंट होंगे तो हमारे पास एक ऑप्टिकली एक्टिव कंपाउंड प्रेजेंट होता है. सो एक्सेप्ट फॉर ग्लाइसिन ऑल द अदर नेचुरली ऑकरिंग अमीनो एसिड्स विल बी ऑप्टिकली एक्टिव. मोस्ट नेचुरली ऑकरिंग अमीनो एसिड्स हैव L configuration. L amino acids are represented by writing the NH2 group on left hand side. Usually LD अब हमें पता है is nothing to do with rotatory activity, but L will mean that the amino group will be on the left side. So usually you will come across L amino acids only. Now what is the structure of proteins? Of course they are polymers of amino acids. You've already read that the proteins are polymers of alpha amino acids and they're connected to each other by peptide bond or peptide linkage. Chemically, the peptide linkage is an amide formed between COOH group and NH2 group. एक amide बनेगा यानी CONH. यहाँ से CO आएगा, यहाँ से NH आएगा. Removal of water molecule से peptide bond formation takes place. ठीक, एक का carboxylic group, दूसरे का amino group. The reaction between the two molecules of similar or different amino acids proceeds through the combination of the amino group of one molecule with the carboxyl group of the other. तो एक का amino आएगा, दूसरे का carboxyl group आएगा. This results in the elimination of a water molecule and formation of a peptide bond. यही होता है peptide bond. Basically occurs between the carboxylic group and the amino group. The product of the reaction is called a dipeptide because it is made up of two amino acids. For example, when carboxyl group of glycine combines with the amino group of alanine, we get a dipeptide glycylalanine. You can see glycylalanine किस किस का बना है, glycine और alanine का. So moving on, we have If now a third amino acid combines to a dipeptide, product को tripeptide बोलेंगे, contains 3 amino acids, easily linked by 2 peptide linkages. Similarly, when 4, 5 or 6 are linked, the respective products are tetrapeptide, penta or hexapeptide. When the number of such amino acids is more than 10, we call them polypeptides. तब हम tetra, penta फिर खत्म कर देते हैं. 10 से ज़्यादा हो तो polypeptide बोल देंगे, वरना oligopeptide भी. A polypeptide with more than 100 amino acid residues having molecular mass more than 10,000 units is called a protein. तो protein is not a polypeptide. ये एक बहुत बड़ा polypeptide है जिसमें more than 100 amino acid will be there. However, the distinction between a polypeptide and a protein is not very sharp. Polypeptides with fewer amino acids are likely to be called proteins if they ordinarily have a well-defined conformation of a protein, such as insulin, which contains 51 amino acids. So, ज़रूरी नहीं है कि 100 से ज़्यादा हो. Insulin is a protein containing only 51 amino acids, yet we call it a protein. अगर इसके पास protein जैसा conformation है. Proteins can be classified in two types on the basis of their molecular shape. एक तो हमारे पास fibrous proteins आएंगे. अब fibrous proteins क्या है? Chain when the polypeptide chain runs parallel and are held together by hydrogen and disulfide bonds, then fiber-like structure is formed. Such proteins are generally insoluble in water. Some common examples are keratin, present in hair, wool, silk, and myosin, which is present in muscles. Fibrous protein में क्या है? Fibrous appearance, fiber-like structure बनेगा. The polypeptide chains will run in parallel to each other. और बीच में क्या बनेंगे? Disulfide bonds बनेंगे. That's how they'll be attached. Then you have globular proteins. Alright, the structure results when the chains of polypeptide coil to give a spherical shape. अगर वो coil हो गए, sphere बनाया, तब जाके हमारे पास globular proteins आते. These are usually soluble in water. Insulin and albumins, they are the common examples of globular proteins. Wool, silk वगैरह, keratin hair protein जो है, वो हमने देखा fibrous थे. यहाँ पे albumins, insulin, they are globular proteins. वो तब बनते हैं जो polypeptides coil around. Alright, structure and shape of proteins can be studied at 4 different levels. ये आपने बायो में अगेन किया है, the primary, secondary, tertiary and quaternary level. हर एक लेवल हम यहाँ पे डिस्कस करेंगे. We have a primary structure of proteins. Primary मतलब length में, along the length of the polypeptide chain, what are the amino acids is composed of? That is what we do in primary structure. Proteins may have one or more polypeptide chains. Each polypeptide in a protein has amino acids linked with each other in a specific sequence, and in this it is this sequence of amino acids that is said to be the primary structure of that protein. Any change in this primary structure, the sequence of amino acid, creates a different protein. तो हमारे पास primary structure में along the length of, हमारे पास बहुत सारी ऐसे lengths हो सकती है, बहुत सारी chains हो सकती हैं, उनको आपस में फिर हम बाद में bond करते हैं. But on the primary level, we just consider one chain and the amino acids it is composed of. What is now the secondary structure? This refers to the shape in which a long polypeptide chain can exist. किस shape में अब जो हमारा chain बना amino acid से वो कैसे exist करेगा? Either it's a helix, ठीक है, बीटा प्लीटेड शीट, दो हमारे पास यहाँ है, हेलिक्स और बीटा प्लीटेड शीट. ठीक, ये हेलिक्स यू कैन सी राइट, ऐसे गोल गोल, just like DNA we did this. These structures arise due to the regular folding of the backbone of the polypeptide chain due to hydrogen bonding between the carbonyl and the NH groups of the peptide bond. Alpha helix is one of the most common ways in which a polypeptide chain can form all possible hydrogen bonds by twisting into a right-handed screw or helix with the NH group by each amino acid residue hydrogen bonded to the carbonyl of an adjacent turn of the helix. Very easy. Alpha helix यहाँ पे दिया हुआ है, ये हमारे पास सेकेंडरी स्ट्रक्चर में पहला एग्जांपल है. दिस हाइड्रोजन बॉन्डिंग बिटवीन द कार्बोनिल ग्रुप, और NH ग्रुप का जो H है वो मोर इलेक्ट्रोनेगेटिव ऑक्सीजन के साथ हाइड्रोजन बॉन्डिंग करेगा. ठीक है, अल्फा वे में इन अ राइट हैंडेड फैशन दिस इज गोइंग टू कॉइल, नॉट कॉइल अराउंड, दिस इज गोइंग टू बॉन्ड इन दिस वे. अब हमारे पास दूसरा आता है बीटा प्लीटेड शीट. इसमें क्या हो रहा है? दिस इज अ बीटा प्लीटेड शीट, डायग्रामेटिकली भी आप देख सकते हैं. It structure, all peptide chains are stretched out to nearly maximum extension and then laid side by side, which are held together by intermolecular hydrogen bonds. Structure resembles the pleated folds of drapery and therefore is known as beta pleated sheet. Pleated saris, you have those drapes जिनको आप pleating करते हैं, तो ये वैसा appearance देता है. I don't know if you can actually make it out कि वैसा ही है या नहीं. तो this happens basically. Alright, I think I'm messing it up a little. I'm just going to erase this. So I just hope you're able to grasp how it is in a pleated manner. But इसमें क्या होता है, जितने भी polypeptide chains हैं उनको हम to its maximum सबको stretch करेंगे और फिर जो side by side linkages बनेंगी, ठीक है, this was ऊपर नीचे, यहाँ पे side से chains बनेंगी, hydrogen bonds again, intermolecular hydrogen bonds. Now, moving on to tertiary structure of proteins. Represents overall folding of the polypeptide chains, that is further folding of the secondary structure. ठीक है, अब secondary structure further fold करेगा, उसको tertiary structure बोलेंगे. It gives rise to two major molecular shapes, fibrous and globular, जो हमने already देखा. Main forces which stabilize the 2° and 3° structures of proteins are hydrogen bonds, disulfide linkages, van der Waals and electrostatic forces of attraction. These are all the bonds जो उनको stabilize करते हैं. Quaternary structure now. Some of the proteins are composed of two or more polypeptide chains referred to as subunits. The spatial arrangement of these units with respect to each other is known as quaternary structure. यानी एक से ज़्यादा दो या दो से ज़्यादा polypeptide chains जो हैं वो फिर sub units बनाते हैं. And how are they arranged in space? उनका spatial arrangement will give you the quaternary structure of the protein. Now you have certain quaternary structures. You have a primary structure, उसके बाद secondary structure दिया है, फिर tertiary कि secondary आपस में कैसे coil कर रहा है, और फिर quaternary हमने दो या दो से ज़्यादा polypeptides लिए, वो आपस में कैसा bonding दिखा रहे हैं. Alright. So again something really colorful. You have a primary structure given, carbon, hydrogen. Then you have a secondary structure जहाँ पे N और O के बीच में hydrogen bonding होगा. You can have then the tertiary structure and the quaternary structure. आपस में two different polypeptide chains bonding आपस में. ठीक. You have now denaturation of proteins. छोटा सा topic है. Protein found in a biological system with a unique 3D structure and biological activity is called a native protein. Native protein क्या है अब? जिसका एक unique structure होगा और जो active होगा. When a protein is in its native form, it's subjected to physical change like change in temperature or chemical change like change in pH. The hydrogen bonds are disturbed. Due to this, globules unfold and helix get uncoiled and protein loses its biological activity. And this is called denaturation of protein. अगर आपने conditions को इतना change किया, you have changed the temperature, you have changed the enzymatic environment around, you have changed the pH, hydrogen bonds disturb होते हैं और जो उनका वो जो हम तब से structures पढ़ रहे थे, there for a reason. और अगर वो structures अनवाइंड हो जाएंगे तो प्रोटीन्स का जो एबिलिटी है, जो भी उनका काम है, दे वोंट बी एबल टू फंक्शन प्रॉपर्ली. उसी को डीनेचुरेशन बोलते हैं. सेकेंडरी एंड टर्शियरी स्ट्रक्चर्स आर डिस्ट्रॉयड बट प्राइमरी स्ट्रक्चर्स रिमेन इंटैक्ट. दिस इज वेरी इंपॉर्टेंट टू रिमेंबर. अमीनो एसिड्स को तो नहीं खोलेंगे ना डीनेचुरेशन में. हेलिसेस अनकॉइल हो सकते हैं या ग्लोब्यूल्स खुल सकते हैं, लेकिन अमीनो एसिड का चेन विल रिमेन इंटैक्ट. कोगुलेशन ऑफ एग वाइट ऑन बॉइलिंग इज अ कॉमन एग्जांपल ऑफ डीनेचुरेशन. अनदर एग्जांपल इज कर्लिंग ऑफ मिल्क, व्हिच इज कॉज्ड ड्यू टू द फॉर्मेशन ऑफ लैक्टिक एसिड by the bacteria present in milk. Moving on to enzymes. Very important again and easy also. Life is possible due to the coordination of various chemical reactions in living organisms. An example is the digestion of food, absorption of appropriate molecules, and ultimately production of energy. This process involves a sequence of reactions, and all these reactions occur in the body under very mild conditions. This occurs with the help of certain biocatalysts called enzymes. All the enzymes are globular proteins almost. Enzymes are very specific for a particular reaction and for a particular substrate. They're generally named after the compound or class of compounds upon which they work. For example, the enzyme that catalyzes hydrolysis of maltose into glucose is named as maltase. So enzymes are very important. Our body cannot carry on its biochemical reactions without the biochemical catalysts, the enzymes which catalyze the reactions. Alright, so for that we have enzymes. They can be proteins, they can be RNAs also. So that's how our life goes on. And they're very specific because for a certain substrate, there'll be only a certain enzyme that will be working for it. So say, for example, you have maltose over here. It's getting converted into glucose and you need a specific enzyme for it, and that is maltase. Sometimes enzymes are also named after the reaction where they're used. For example, the enzymes which catalyze the oxidation of one substrate with simultaneous reduction of another substrate are named as oxidoreductase enzymes. So what happens is basically enzyme ने एक चीज को oxidize किया, दूसरे को reduce किया. That's where oxidoreductase enzyme would come out. So after the reaction, you're naming the enzyme. The ending of the name of an enzyme is ase. Mechanism क्या है? Enzymes are needed only in small quantities for the progress of a reaction. Similar to the action of chemical catalysts, enzymes are said to reduce the magnitude of activation energy. What do catalysts do? We call enzymes as the biocatalysts. Catalysts reduce the activation energy, provide an alternate path for the reaction which is easier. That's what the enzymes do too. For example, activation energy for acid hydrolysis of sucrose is this, while the activation energy is this only. इतना कम हो रहा है, 6.22 से 2.15 when hydrolyzed by the enzyme sucrase. So mechanism for the enzyme action has been discussed later. We have now vitamins. It has been observed that certain organic compounds are required in small amounts in our diet, but their deficiency causes specific diseases. These compounds are called vitamins. Most of the vitamins cannot be synthesized in our body, but plants can synthesize almost all of them, so they're considered as essential food factors. However, the bacteria of the gut can produce some of the vitamins required by us. All the vitamins are generally available in our diet. Different vitamins belong to various chemical classes. It's difficult to define them on the basis of structure. They're generally regarded as organic compounds required in the diet in small amounts to perform specific biological functions for normal maintenance of optimum growth. So usually they cannot be and health of the organism. Usually they cannot be considered to be a main part of the diet, but yes, if they're deficient, then you can see certain deficiency disorders there for sure. Vitamins are designated by alphabets A, B, C, D, etc. Some of them are further named as subgroups: B1, B2, B6, B12. We know it, right? Excess of vitamins is also harmful. Vitamin pills should not be taken without the advice of a doctor. The term vitamin was coined from the word vital plus amine, since the earlier identified compounds had amino groups. Later work showed that most of them did not contain amino groups, so the letter E was dropped and the term vitamin is used these days. So basically vital amines, important amino groups. So earlier amino acids say amino groups were found in these vitamins, so they were called vitamin. Eventually हमें पता चला कि सब में amino groups नहीं होते, then we replaced the E, we just removed it, so we have vitamin right now. Now what is the classification? Vitamins are classified into two groups depending upon their solubility. या तो वो fat soluble होंगे या फिर they're going to be water soluble compounds. Vitamins which are soluble in fat and oils but insoluble in water are kept in this group. Self-explanatory term है. They're vitamin A, D, E and K. ये आप याद रखेंगे कौन कौन से हैं. They're stored in liver and adipose fat storing tissues. Now water soluble, you have B group vitamins and C are soluble in water, so they're grouped together. Water soluble vitamins must be supplied regularly in diet because they're readily excreted in urine and cannot be stored, except vitamin B12 in our body. Some important vitamins, their sources and diseases caused by their deficiencies are listed over here. This usually tends to come vitamin A, but it's really easy. Sources, ye sare hai: fish, liver oil, and xerophthalmia, the hardening of cornea, night blindness. Then B1, thiamine, B2, riboflavin, B3, B6, pyridoxine, vitamin B12, pernicious anemia, again very important. Vitamin C, ascorbic acid, scurvy hota hai, bleeding gums, right? Yes. Then you have vitamin D, rickets, again important, exposure to sunlight, fish and egg yolk. Then vitamin E, increased fragility of RBCs. Vitamin K, increased blood clotting time. So now moving on to nucleic acids, you can go through the table on your own also about the deficiency diseases of vitamins. Now we have nucleic acids. Every generation of each and every species resembles its ancestors in many ways. So they're talking about genetics, right? How are these characteristics transmitted from one generation to the next? It has been observed that nucleus of a living cell is responsible for this transmission of inherent characters, also called heredity. The particles in nucleus of the cell responsible for heredity are called chromosomes, which are made up of proteins and another type of biomolecule called nucleic acids. These are mainly of two types. We have DNA and we have RNA, deoxyribonucleic acid and ribonucleic acid. Mainly यही nucleic acids है जो हमारी body comprises. Since nucleic acids are long chain polymers of nucleotides, so they're also called polynucleotides, all right? Nucleotides के बने हुए होते हैं. Nucleotide in itself is containing of a nitrogen base, a phosphate group and a ribose sugar. Over here it can be deoxyribose or it can be just ribose, which is basically a pentose sugar, and they are responsible for inheriting all the information from parents to the offsprings. Now what is the chemical composition? We have done this in detail in biology. Complete hydrolysis of DNA or RNA yields a pentose sugar. Phosphoric acid and nitrogen containing heterocyclic compounds called bases, because these are the components. In DNA, the sugar moiety is beta D2 deoxyribose, whereas in RNA it is beta D ribose. So it's beta sugar, it's D, and it's ribose in case of RNA, and it's deoxyribose in case of DNA. Fine. So this is the beta D ribose. We know beta किसके base पे होता है? Anomeric carbon, right? D and L based upon where do you have, whether it's on the left or on the right. That is D and L. They denote the position of specific groups in a compound. DNA contains 4 bases: adenine, guanine, cytosine, and thymine. Fine. These are the 4 different types of nitrogen bases present. They can be purines or pyrimidines. RNA also contains 4 bases. The first 3 bases are same as in DNA, but the fourth one is uracil. So पहले 3 सब में same है, but thymine will be in DNA and uracil will be in the RNA. That's the only difference in case of nitrogen bases. Now what is the structure? A unit formed by the attachment of a base to 1' position of sugar is known as nucleoside. So we get sugar plus base as a nucleoside. The sugar atoms are numbered as 1', 2', 3'. In order to distinguish these from the bases, when nucleoside is linked to phosphoric acid, we get a nucleotide. So nucleoside is nitrogen base plus sugar. If you add a phosphate moiety to it, we get a nucleotide. So alright, this is a nucleoside pehle, baad mein when you attach the phosphate group, we get a nucleotide. Nucleotides are joined together by phosphodiester linkage between 5' and 3' carbon of the pentose sugar. Alright, 5' and the 3' carbon. So there will be phosphodiester linkage. The formation of a typical dinucleotide is shown over here. So here we go. This is a sugar. This is a sugar, this is a nitrogen base, this is a nitrogen base, this is the phosphate group, this is the phosphate group. When you are trying to link them, what happens is, this phosphodiester, you have ester, sugar basically, the oxygen and phosphate group ka oxygen and this carbon, they're going to form a bond. This is phosphodiester linkage. A simplified nucleic acid chain is as shown below. You have a sugar attached to a base, then a phosphate, then again a sugar, phosphate, base, sugar, phosphate. So basically the backbone is the sugar phosphate chain, and what protrudes out is the bases. Information regarding the sequence of nucleotides in the chain of a nucleic acid is called its primary structure. Nucleic acids have a secondary structure also. James Watson, Francis Crick gave a double-stranded helix structure for DNA. Watson and Crick model, it's basically the secondary structure. Primary structure is just the nucleotides. Alright, sequence of the nucleotides. Two nucleic acid chains are wound about each other and held together by hydrogen bonds between pairs of bases. The base pairing ATGC, double and triple hydrogen bonds respectively, that we've already discussed in bio. That's what it's talking about. The Watson and Crick model, wherein you talk about the double helix structure. The two strands are complementary to each other. Tabhi jaake hydrogen bonds are going to be formed between specific base pairs. Adenine will form a hydrogen bond with thymine. Cytosine will form hydrogen bond with guanine. Alright. Then we have secondary structure of RNA. Single-stranded helix is present. RNA single-stranded, which sometimes folds back on itself. RNA molecules are of 3 types and they perform different functions. They are mRNA, messenger one, then the rRNA, ribosomal, and the tRNA, which is the transfer RNA, and they have their own specific functions. Alright. Now you have a little paragraph given about DNA fingerprinting. It's known that every individual has unique fingerprints. These occur at the tips of the fingers and have been used for identification for a long time. In forensics, but these can be altered by surgery. A sequence of bases on DNA is also unique for a person, and information regarding this is called DNA fingerprinting. It's same for every cell and cannot be altered by any known treatment. So DNA fingerprinting, why did it come into play? Because original fingerprinting, by surgical methods, fingerprints can be changed. But then if you have DNA fingerprinting, if you tally the base pairs, they're always unique for a person. So that becomes a better way. In forensic laboratories for identification of criminals, to determine paternity of an individual if there is a dispute, to identify the dead bodies in any accident by comparing the DNAs of parents or children, then to identify racial groups, to rewrite biological evolution. Then you have biological functions of nucleic acids. DNA is the chemical basis of heredity and may be regarded as the reserve of genetic information. Transmission, RNA can do well, but if you have to store genetic information, DNA is the better option. DNA is exclusively responsible for maintaining the identity of different species of organisms over millions of years. A DNA molecule is capable of self-duplication during cell division, and identical DNA strands are transferred to daughter cells. Another important function of nucleic acid is the protein synthesis in the cell. The central dogma, DNA to RNA and RNA to protein. This becomes a function again. Actually, the proteins are synthesized by various RNA molecules in the cells, but the message for the synthesis of a particular protein is present in the DNA only. So DNA will send the message to RNA, though the proteins will be formed by RNA later on. So these are all the functions: storage of genetic information, self-duplication, protein synthesis, etc. Now moving on to hormones. They're molecules that act as intercellular messengers. What transmits information between two cells? These are produced by endocrine glands in the body, poured directly in the bloodstream because they they have no ducts. In terms of chemical nature, some of these are steroids, example estrogens and androgens. Some are polypeptides, for example insulin and endorphins, and some others amino acid derivatives such as epinephrine and norepinephrine. They have various origins. Hormones have several functions in the body. They help to maintain the balance of biological activities in the body. You might have done an endocrinology in biology also. They're very important for maintaining, coordinating all the functions inside the body. Say for example, role of insulin in keeping the blood glucose level within the narrow limit is an example of this function. It's released in response to the rapid rise in blood glucose level. On the other hand, we have glucagon, which will counteract. It will tend to increase the glucose level in the blood, whereas insulin will try to decrease this rate. The two hormones together regulate the glucose level in the blood. Epinephrine and norepinephrine, again, they mediate responses to external stimuli. Growth hormones and sex hormones play a role in growth and development. Then you have thyroxine in the thyroid gland. It is an iodinated derivative of amino acid tyrosine. Tyrosine को iodinate करेंगे, you will get thyroxine. All right. Abnormally low level of thyroxine leads to hypothyroidism, which is characterized by letharginess and obesity. Increased level of thyroxine causes hyperthyroidism. Low level of iodine in the diet may lead to hypothyroidism because you need to iodinate the what did we see over here? You have to iodinate tyrosine, which is an amino acid. So if you have low levels of iodine, hypothyroidism will pertain. And what we have is largely being controlled by adding sodium iodide to commercial table salt that is known as iodized salt. So this will help to ensure that all of the individuals take in good amount of iodine so that hypothyroidism is prevented. Then we have steroid hormones produced by the adrenal cortex and the gonads, testes in males and ovaries in females. Hormones released by the adrenal cortex play very important role in the function of the body. तो जो adrenaline वगैरह जो भी release होते हैं, all the steroid hormones from the adrenal cortex, they're very important. For example, glucocorticoids, they control the carbohydrate metabolism, modulate inflammatory reactions, and are involved in reactions to stress. Then the mineralocorticoids, they're all released by adrenal cortex, control the level of excretion of water and salt by the kidney. If adrenal cortex does not function properly, then one of the results may be Addison's disease, all right, characterized by hypoglycemia, low level of sugar, weakness, increased susceptibility to stress. The disease is fatal unless it's treated by glucocorticoids and mineralocorticoids. So carbohydrate metabolism, producing glucose inside the body, glucocorticoids do it. Inflammatory reactions, even reaction to stress. Then mineralocorticoids, the level of excretion of water, how much of salt, how much of mineral is to be kept in the body, how much is to be excreted out. Mineralocorticoids are going to look after that. Hormones released by gonads are responsible for the development of secondary sexual characters. Testosterone is the major sex hormone produced in males. It is responsible for development of secondary male characters, say a deep voice, facial hair, general physical constitution. And estradiol is the main female sex hormone, all right, estradiol, and it's responsible for development of secondary female characteristics and participates in the control of menstrual cycle. Progesterone is responsible for preparing the uterus for implantation of fertilized egg. All of these are your different hormones in the body and playing different roles. So in biology we do this in great depths, but here you just have to look at them at a surface level. So with that we come to an end of the lesson. I hope you enjoyed the lecture. If your basics are done, this can be the best go-to video lecture that you can watch. Thank you so much for watching. I'll see you in my next one.